Welcome to the ultimate security system on Earth. Your cells. Right now, trillions of tiny invaders are trying to break inside you. Every living cell is an empire, and like any empire, its survival depends entirely on border control. This is the plasma membrane. It isn't a rigid wall, it's a dynamic, self-healing matrix known as the fluid mosaic model. Its backbone is a phospholipid bilayer, amphipathic molecules with hydrophilic, water-loving heads facing outward, and hydrophobic, fatty acid tails shielded on the inside. Scattered within this sea are cholesterol molecules, acting as temperature buffers to keep the membrane from freezing or turning completely liquid. Embedded throughout are integral and peripheral proteins. Together, they create a barrier that determines what enters the cellular economy and what stays locked outside. The fluid mosaic model describes the structure of the cell membrane as a dynamic, flexible bilayer of phospholipids embedded with a patchwork of proteins, cholesterol, and carbohydrates. First proposed by S.J. Singer and Garth L. Nicholson in 1972, it remains the most widely accepted scientific model for explaining how plasma membranes look and function. The term fluid signifies that the individual lipid and protein molecules are not rigidly fixed and can drift laterally within the plane. The term mosaic refers to the colorful variety of molecules scattered across the membrane like tiles in an artwork. In biology, a concentration gradient results from the unequal distribution of particles, e.g. ions, between two solutions, i.e. the intracellular fluid, the solution inside the cell, and the extracellular fluid, the solution outside the cell. This imbalance of solutes between the two solutions drives solutes to move from a highly dense area to a lesser dense area. This movement is an attempt to establish equilibrium and eliminate the imbalance of solute concentrations between the two solutions. The membrane operates on a fundamental law of physics, entropy. In passive transport, molecules move down their concentration gradient from areas of high concentration to low concentration, requiring absolutely zero cellular energy. Small, nonpolar molecules like oxygen and carbon dioxide dissolve right through the hydrophobic core via simple diffusion. Simple diffusion is the passive movement of molecules from an area of higher concentration to an area of lower concentration. It requires no cellular energy, ATP, and relies solely on the natural, random kinetic motion of particles until they reach an even state of balance, known as equilibrium. But what about polar molecules like water? Because water is repelled by the fatty acid tails, it utilizes specialized molecular highways called aquaporins. Aquaporins are specialized channel proteins embedded in cell membranes that facilitate the rapid and highly selective transport of water molecules into and out of cells. Often referred to as plumbing systems for cells, they allow for rapid water flow in response to osmotic gradients while blocking charged ions that would disrupt electrical balances. When larger charged ions need to cross, they use gated ion channels or carrier proteins in a process called facilitated diffusion. It's highly selective, but it's entirely driven by the natural, kinetic drift of the universe. But cells rarely want equilibrium. Equilibrium is death. To survive, cells must constantly fight physics and pump molecules against their concentration gradient. This requires active transport, fueled by the cell's energy currency, ATP. Consider the sodium-potassium pump, NAEK-ATPase. By consuming one molecule of ATP, this molecular machine forces 3 sodium ions out of the cell and pulls 2 potassium ions in. This relentless pumping creates a massive electrochemical gradient, essentially turning your cell membrane into a cellular battery. This biological voltage is what powers your nervous system, allowing your brain to send signals to the rest of your body right now. When the cell needs to move massive cargo, it switches to vesicular transport. In endocytosis, the membrane literally pinches inward to engulf external materials. But this opens a dangerous vulnerability. Pathogens like influenza or HIV have evolved to hijack a highly specific pathway called receptor-mediated endocytosis. The virus features surface spikes that perfectly mimic friendly ligands. They bind to the cell's receptors, essentially tricking the cell into pulling the enemy straight across the border. Once inside, the viral payload is released, turning the cell's own machinery against itself. On the flip side, the cell clears out its own waste and secretes hormones via exocytosis, where internal vesicles fuse with the plasma membrane to vomit contents into the extracellular space. When these transport systems fail, the consequences are lethal. In patients with cystic fibrosis, a genetic mutation disrupts a single active transport protein responsible for pumping chloride ions. Without this ion movement, osmosis fails, water stops flowing, and thick, deadly mucus suffocates the lungs. Understanding the membrane isn't just basic biology, it is the foundation of modern medicine. Every pharmaceutical drug you take is specifically engineered to target, block, or slip past these exact transport mechanisms. From simple diffusion to ATP-driven pumps, your life exists purely because of the complex, microscopic traffic control happening across this 10-nanometer border. Thanks for watching. If you want to dive deeper into molecular biology, hit subscribe and leave your thoughts on the sodium-potassium pump below.